So now that we're studying cellular respiration, let's take a look at how this whole idea of energy coupling relates to that process of cellular respiration. So let's look at where the exergonic part of the process is, and then also where the endergonic process is at. So when we talk about cellular respiration, this is going to be the overall equation that we're looking at. Okay, so when we look at this, it's definitely a summary equation, and we tend to think about the process of cellular respiration as something that's exergonic. And the reason that we think of that is because here we do have energy produced, so it is overall certainly exergonic. But even so, when we run exergonic processes, the whole reason we do that is because we do want to use that energy to do something that is endergonic, or something that is going to require energy. So this energy that's produced here as a product of this process, what that's going to be used for is that's going to be used to make ATP. And so that right there will be the endergonic process. And if we just talk briefly about how we're going to make that ATP, we're going to have two overall processes that we use to make the ATP. We will have substrate level phosphorylation. That's how we'll make about 10% of the ATP that we do generate during this overall process. But the vast majority of the ATP is going to be made by the process of chemiosmosis. So we will see some endergonic processes in the first two stages to make the ATP by substrate level phosphorylation. So again, that's by glycolysis or during glycolysis, and then also the citric acid cycle. And then the chemiosmosis part, our second way of making ATP, that's going to generate about 90% of the ATP that we make, and that will be in the third stage, which is our oxidative phosphorylation. Okay, so overall, again, while cellular respiration is exergonic and we do have a net release of energy here, we do want to know that the whole reason we're running this exergonic process is because we want to take that energy and we want to use it for an endergonic process. And that endergonic process is going to be the actual synthesis of ATP. Two different methods down here that we'll use to do that. We will have substrate level phosphorylation. We'll also have chemiosmosis. And altogether, those are going to generate about 38 ATPs per glucose that we start with.